We'll get started then. I'm Paul Mirabelle. I'm chairman of the Research Support and Prevention Committee at Tour de Cure, and we'd like to welcome everyone to our fourth webinar of the series, Cancer Doesn't Sleep and Neither Do We. As always, raising funds for cancer research and prevention remains our #1 priority, but as a part of this program, though, we're introducing the community to our research colleagues who are deeply embedded in doing the work which is so important. And I'm joined today by Associate Professor Michael from Flinders University, who was recently appointed as the lead of cancer research program in the new Flinders Health and Medical Research Institute. A bit of background on Michael. After helping to establish one of Australia's first biotech companies, Michael moved to Flinders in 1998 in the pursuit of biomarkers for colorectal cancer. He pioneered global profiling of microRNAs to identify associations with disease, particularly solid tumors. His interests have broadened since then to investigate the involvement of epigenetics, and he might tell us a little bit about what that means, and non-coding RNAs in various aspects of human development and disease. However, the focus of his lab is still to deliver diagnostic tests that will inform the clinical decision-making and to help identify drug targets for novel therapeutics. Michael contributes to community and regulatory bodies, including the Gene Technology Technical Advisory Committee of the Office of the Gene Technology Regulator. He has been the past recipient of Tour de Cure grants. We're absolutely delighted that Michael could join us today. First of all, Michael, let me ask you, how are you? And as I've asked the other researchers, how are you coping in this COVID-19 world?
Hi, Paul. I'm well, thank you. So welcome to Flinders, virtually. It's an interesting time, isn't it? But it's been in some ways a frustrating time, but also kind of an affirming time as well for people, I think. It's asked us to question what's important in our lives and in society. It's forced us to reevaluate our connection with the health system and public health. So I'm well. It hasn't disrupted me too much. I had a couple of family members who came from overseas and had to hold up at home for a short time till they got a test result. Yep. But all's well, thank you.
Very good. How's the lab functioning under the isolation rules that we've had as a country over the last 8 weeks or so?
Yeah, so my lab actually hasn't been disrupted all that much. The Flinders University has had a policy of keeping the labs going, do as much work at home as you can, but the laboratory work, they just kept that running. And we're lucky, we've got nice big labs, lots of space, so we can do it safely. So that's worked well. I actually work for the hospital here, believe it or not, Flinders Medical Centre. Yeah. And of course, that's been all hands to the pump. And we've been very much a part of the COVID action here in Adelaide. We have a COVID testing clinic right next to the cancer centre here.
Terrific. The actual centre there, it's termed an innovation centre. How do you define that? How does that set the parameters for the work that you have underway?
Yeah, so I guess it's a bit of a cliche, but it is thinking outside the box. It's trying to address things in a slightly different way. We have quite a good history in that here at Flinders. We're the first to introduce digital PCR, believe it or not. Pam Sykes and Alec Morley here, quite a few years ago now, and that's now a really big thing in the diagnostic space.
When you say, let me pause you there for our audience. When you say digital PCR, what are you referring to?
I'm referring to a way of measuring molecules, nucleic acids, DNA or RNA, at really small levels to do things. So they first developed this so they could pick up sort of the residual disease of blood cancers in the blood.
Right.
Yeah. Okay. But on top of that, we see ourselves as being innovative, doing things like developing new blood tests for colorectal cancer. One of our team was part of a Eureka Prize a few years ago. So we've been trying to push ourselves at that limit. We were first developed as a cancer centre by a chap called Graham Young, who helped introduce the National Bowel Cancer Screening Program to Australia, actually. And so cancer prevention, cancer screening, those sorts of things are really part of our DNA here, very important to us. Also cancer survivorship, which are things that aren't that important in a lot of other, lab-based cancer research centres. But on top of that, we still do a lot of clinical trials and a lot of really excellent lab-based work as well.
One of the, what you're identifying is basically areas of particular focus that you take on board there at Flinders. And I think, for our supporters and our corporate partners and so on, they will not necessarily have an understanding in terms of how different institutions frame their work and prioritize their work. Give some insight. How did that happen? Like what, if you think about the areas of focus that Flinders decided that you're going to be very good at and you're going to devote your attention at, because there's any number of issues which are outstanding when it comes to cancer research. How does that get set? How does that get modified over time?
Well, primarily, I think it reflects the fact that we operate within a hospital, and that what we try to reflect the clinical excellence that we have here. So areas of real clinical strength often are the ones that will emerge as research strength as well. So we build on the clinical strengths that we have. The example I said before, the chap who set up the bowel cancer screening program was the head of gastroenterology. And he worked very well with the head of surgery as well. So for that reason, we have strengths in GI cancers from top to tail. And so we are trying to develop new diagnostic tests for that, but also therapeutics and a variety of other things that I'm sure we're going to get to.
Yeah. You've been the recipient of a number of grants from Tour de Cure. And one of the things that we're very keen to do, which is why I'm delighted that you could join us today, is to close the loop, if you will, between our riders, our volunteers, our corporate partners and their employee teams that have been so instrumental in raising funds. The more they understand about what happens on the research side of things and actually personalizes the research efforts, the better it is for everyone. So, let me ask you a couple of questions around those grants. First of all, without getting into the detail of the grants themselves yet, how important are the efforts of groups like Tour de Cure and other fundraisers when it comes to funding cancer research?
Look, absolutely essential these days. The research dollar hasn't really risen all that much at the NHMRC level for many years. It's stayed the same. And that's
that's the main Commonwealth government funding body when it comes to medical research.
Correct. Yeah. And so philanthropic funds are the lifeblood of many labs and institutes. And they provide us the ability to generate a lot of preliminary data that we can then go to one of the big grant bodies and get much more. So in that way, you're leveraging your investment as a philanthropic body, which is really important. Of course, now the government's introduced the new MRFF fund, and they target that specifically. So having broader philanthropic activity is really important. So that way you can keep generating activity so that when those other things become important, then you go to the MRFF. That's the Medical Research Future Fund that the government's brought into play.
Right.
Yeah.
So take me through the research that you've been doing, which Tour de Cure has funded part of it and so on. And maybe assume for a second, Michael, that you're dealing not necessarily with a colleague, but you're dealing with your mother. Sure. And tell us what you're doing, and tell us what sort of insights are emerging from that work, if you will.
Okay. So you asked about epigenetics before. So yeah. I'll just explain what that term means. As most people know, we have a genome, the DNA that makes up our cells. And all our cells have that same DNA. And of course, it's different cells have different functions. Brain cell from a liver cell. So they have different genes that are active. And so there are mechanisms that activate or inactivate genes. We've got about 20,000 genes all up. And in any one particular cell type, there's maybe 2/3 of the genes are active in that cell. And those combinations differ. Okay, so what happens is our genes are activated and inactivated by a series of developmental processes. They're often epigenetic processes. But beyond that, our genome actually has to interact with our environment as well. I work on colorectal cancer. Our gut is constantly exposed to the diet that we have. So for us, epigenetics is really looking at how different diets might predispose us or protect us from developing bowel cancer. And so along that path, early on, we identified really one of the products that we didn't identify, but we worked on one of the products that the bacteria in our gut produce when we eat a really good, healthy, high fiber diet that seem to protect us from bowel cancer because it's the preferred energy source. This product's called butyrate, comes from the Greek word for butter, actually.
Yeah.
Butyrate is the preferred energy source for a normal gut cell. Whereas if you expose it to a cancer cell, they die really quickly. We call that the Butorac Paradox. And we were trying to understand how that worked. And we were looking at these little microRNAs, these little snippets of gene products that actually regulate how other genes work. And we understood that the butyrate phenomenon happens to occur through these microRNAs. That got us interested. Can we use these therapeutically? Can we use these to kill the cancer cells specifically? And beyond that, other people were addressing this butyrate issue. Why does it feed a normal cell but kill a cancer cell? And they came to the conclusion, in fact, that it was because cancer cells produce energy differently. Now, you know, all the Tour de Cure riders know, when you're riding normally and you're sucking in a lot of oxygen, you're going well, okay? There's a way of producing energy with a lot of oxygen. When you start to run out of oxygen, your body starts producing lactic acid in your muscles. They start to feel heavy. So we've got mitochondria that can produce energy very efficiently with oxygen. When there's not so much oxygen, we have another pathway that comes on that produces lactic acid. Two different pathways. So normally mitochondria do the work, but in cancer cells, they tend to like this lactic acid pathway more. So that gives us a bit of an in. That's a real hallmark of cancers, actually. Yeah. It's also the reason PET imaging, if people have a PET scan, the reason a PET scan works is because the cancer cells are sucking in more glucose. So they can burn it with this lactic acid production. So.
And just for the sake, again, for the audience, the PET scan will actually disclose where you have cancerous cells within your body.
Correct. Yep. And the way it does that is because they draw in a labeled glucose form, and that's what you're imaging with a PET scan. And so knowing that, okay, it looks like this dietary thing is metabolic, right, is around producing energy, knowing that our microRNAs are involved in that, we wanted to know if our microRNAs were involved in specifically in the energy production in a cancer cell. And that's the work that Tour de Cure funded for us. And so what we've done is we've been able to go in and look at all these microRNAs. And in fact, we found that cancer cells, if you try and shut down that lactic acid pathway that I mentioned before, they actually cope okay with it. The mitochondria are actually okay. They need this lactic acid pathway to go so they can develop all the other molecules that they need to proliferate quickly, which is what cancers do. So we had to shut down the mitochondria so we could play with this other cancer-specific pathway. And we thought, you know, if we're going to do that, why don't we do it in a really clinically relevant way? And we were lucky because there's a drug that 600,000 Australians take, and they're the diabetics who take metformin. And that's what metformin does. It shuts down your mitochondrial energy production pretty much. So we've used that. We've used metformin, this diabetes drug, to really dissect how cancer cells produce energy and to see which microRNAs are important for that. And we've found now, so the outcome of all of this work that you've funded is that we've discovered microRNAs that actually shut down glycolysis. We've shown other ones that shut down the mitochondrial activity. And now we're trying to bring this all together. And we're trying to understand what genes they regulate, how they regulate energy production, and, you know, maybe in combination with metformin, a very safe drug, whether we can develop some new therapeutics to actually give people. And so that's really exciting. We've also done work with Flinders Foundation, who is a Tour de Cure partner that you know really well. And we've done screens with metformin and libraries of FDA-approved drugs. So we've actually already got some insight into what sorts of drugs might be working there.
What's the impact on the patient today? If someone is diagnosed with a form of cancer, which falls within the remit of what you're looking at. How long before they see changes in clinical protocols in terms of dealing with it? How long does it take to move from the work that you're doing, Michael, to a point where there's actually a clinical protocol which is varied by virtue of that work?
Yes. So I can't give you a time now, but what I can tell you is the strategy that we're using is a repurposing strategy. So it's a strategy that we're trying to use already safe drugs,
yes,
that are FDA approved, and we're trying to repurpose those by combining them and put them in a different context. So in that way, we're hoping that we can deliver a therapy to the patients faster.
No, that's outstanding. Congratulations. And I mean, at the end of the day, you know, the effort that our family and our partners and our corporate supporters put in is all directed towards supporting that sort of work. And it's terrific to hear from you know, a team lead that's actually going through that effort about what is emerging from it. So congratulations.
Thank you, thank you. Can I add something to that quickly?
Absolutely.
So you asked about innovation before and epigenetics. One of the really exciting things for us here is we've recently got a fantastic scientist from Denmark who's joined us in the lab. And she was, she came from Sir Paul Nurse's lab, which won the Nobel Prize for understanding cell cycles, how cells divide. And the way they understood that was using yeast as a model. And she kind of uses yeast as a model for a metastatic cell, for a cell that's splintered off. And in fact, all the early metabolism work was done at Carlsberg Laboratories a long time ago when people were trying to figure out how yeast worked to make better beers.
Yeah.
So she's brought all of this depth of knowledge with her. And this is where the innovation comes in. We can go back to these really early evolutionary creatures like yeast, see what the fundamental biology is that allows a cell to replicate, and then take that and put that in a cancer context. And that is really speeding things up and showing great dividends. So that's a lot of fun too. Scientifically, that's a lot of fun. Yeah.
Michael, you need to learn to become more enthusiastic about your work.
It's hard not to be, right? It's great.
No,
that's why we do it.
No, it's great. It's actually great to see, and it's great for our people to have a chance to see your enthusiasm for what you do. And I have to say, it is a common feature with the researchers that we support. There's a real passion for what you're doing, which, believe it or not, it actually motivates us to work even harder to raise more dollars for it. So it's fantastic to see.
Well, we're a team, right? We're all a team. Yeah.
Absolutely, absolutely, and that is exactly the way that we feel about it. We also feel like we take all of our researchers like you, and whether you like it or not, you're part of the True to Cure family now. So we adopt you as one of our own. So
I've got a lot of friends in True to Cure. It's a great organization.
Tell me this. Well, you know, one of the questions that is often very challenging for lay people that are outside looking in is, you know, how do you define success in research? When you think about your efforts and the timeframes, you know, what are the hallmarks, if you will, of success? It tells you that, yeah, we've made progress here. What are the things that you look to?
Yeah, well, you talk to a university and they'll tell you it's all about publications and citations. I don't think it is. I think it's changing practice, it's changing protocols, it's having a real world effect and helping our patients. Ultimately, it's about helping patients, saving lives, making lives better. That's what we're about. I work in an integrated cancer center. It's designed so that we as researchers walk through the waiting room and we see the patients, and that grounds us. So it's about our patients at the end of the day. So having said that, what's important is, well, for us primarily, identifying some really good diagnostic tests so we can prevent cancer in the first place. I think the best money spent is preventing it. No one thanks you for the cancer they didn't get, but it's a really important part of it.
No, you're quite right.
The cancer prevention and the lifestyle messages, really important. We've got some great teams doing that sort of work here. Beyond that, it's being focused in the lab. And I think because I'm a hospital medical scientist rather than just an academic, it is trying to deliver outcomes. It's trying to deliver new tests. It's creating relationships with people who can get those to market. And we're fortunate to have those relationships. It's also about when you do some really nice basic biology, being able to see, okay, so where's the relevance of this to the patient? How are we going to get this to be a drug? How can we develop clinical trials? We're really lucky here, again, being an integrated cancer centre, we have a really active clinical trials unit. So we've already done some of this work with metformin and repurposing, and some of that's to go into clinical trials for prostate cancer already with other partners.
That's fantastic. What do the next couple of years hold for you?
A lot of excitement, actually. We've recently appointed some new mid-career researchers who are very exciting. And we've got a great deal of depth in that RNA space that I was talking about before. And RNA therapeutics are just on the cusp, the cutting edge. And in fact, COVID has really brought those to the fore. The excitement about a lot of those really early vaccines for SARS-CoV-2, yeah, mRNA vaccines, where you put the mRNA into a cell rather than a bit of the virus. And the mRNA produces the viral protein in your cells, and you develop an immune response to it. So RNA therapeutics are really exciting at the moment, and we're really looking into that space.
I just want to circle back on one of the comments you made that the university does look at publications, for instance, as a key metric of achievement and success. And I suspect we all know why, but I'd be interested, why is it so important in your view? What does publication indicate to the research community?
Well, from a university perspective, that's how they're funded. The governments fund them on their on particular rankings, and those rankings come back to how many publications they have in particular journals. So it's a quirk of the system. You might argue that it's the best way to fund universities or not, potentially.
Yeah.
But that's important. And these are the ERA rankings that I'm talking about, the university rankings. And those rankings actually drive where students want to study. And so you can understand why universities are really geared towards publications. And it's a very academic mindset, and that's fine. It delivers to the general body of knowledge that the world has, and that's a good thing.
Yes.
But at the end of the day, we want it to help a patient too, right?
Yeah, absolutely. I mean, that's just the reality of any organization, isn't it?
Absolutely.
Yeah. If I were to ask you, what are the bottlenecks in terms of generating insights? What's stopping us from moving as a research community faster through this whole process?
Look, the main limitation is it's a resource limitation, and that's where Decoded is fantastic. It's money. It actually is. Beyond that, I think there are some issues around not just research, but the development side. But Australia's getting better at that now. And we've got some good venture capital funds that are coming in to help. And we're starting to develop a culture of entrepreneurship that I think is important for us. We're seeing, companies like CSL being some of the most successful companies in the country. And we all need to build on that, really. So if there are bottlenecks, at the basic research level, it's about funding. It's about being able to keep re-employing the people you've spent so long training,
yeah,
and keeping them in a job and maintaining that corporate knowledge so you can take the next step and the next step and the next step. It's about developing early cancer researchers and young talent. That's fundamentally important. I can say the Tour de Cure grant that you gave me developed two fantastic PhD students. They're now both off doing their first postdocs, and they are wonderful people. So many things will come from that research, but not least the fact that you've set two really intelligent young women on their way to a research career.
That's actually terrific to hear. It's very heartwarming to hear. It's one of the things that we're constantly looking at ourselves to say, how do we demonstrate to everyone that works so hard about what the outcomes are? And we certainly look at publications, but we're actually very explicit that we don't want to limit it to that. And you've already identified a couple of things, but if we can fund some research which allows further research funds to be gained, and we can play the role of an accelerant, if you will, that's a good thing. If we can help young researchers get off the ground and make sure that they get that funding and so on. And as a result, over the last number of years, we've broadened the scope of things. So we have some PhD scholarships now. We have obviously early career, and we introduced a mid-career grant, specifically at critical points to try and help our research community step up to the next mark. So it's actually very reassuring to hear it's having the desired effect.
Yeah, it's having an enormous effect. And I want to thank you collectively from everyone at Flinders, because you've been very helpful to us.
Well, thank you. It's absolutely our pleasure. A couple of other areas I just want to ask you about, since I've got someone that can comment on that. I've noticed, as I'm sure everyone has who's been watching television over the last number of weeks, there's been a renewed push from the Commonwealth government to get people that are eligible for the bowel screening test to participate in that. Take the test, I think is the tagline. Can you just maybe add some of your clinical weight behind that? Why is this so important?
Well, it's fundamentally important because it saves lives. At the end of the day, we know it saves lives. Again, some of the earliest clinical trials were done here before it was rolled out at a national level. I personally remember going to the surgeons and getting a bucket with someone's resection taken to a pathologist so we could take a sample for research, and seeing in someone's cecum, in their appendix, that there was something the size of a cricket ball. He had no idea that that was there.
Yeah.
It saved his life. And from that moment on, I was convinced that this was worth its weight in gold. And it is. So at the moment, we're around 40% compliance. 40% of people are doing the test. Even that is economically viable. I mean, the government wouldn't roll this out if there wasn't a money saving at some stage, right?
Sure.
So even that is paying its way. However, we want to do better because we want to save lives. That's part of the reason we're trying to develop blood tests, for what it's worth, because a lot of people, for whatever, it's a really easy test to do, right? You put your hand in the toilet bowl, you touch nothing, you put it in a test tube, it's very clean, but for some reason that turns people off. They tell us they prefer to go to a GP and do a blood test. So, okay, we're trying to develop some blood tests for them. That's part of what we're doing at the moment.
Yeah.
Yeah. So, I think as people get used to it as well, though, and as we've got young people coming through the phase of middle age and they see their parents doing it, I'm expecting that the compliance is going to go up. But we're trying to also deliver some novel tests that might be more agreeable to people.
One last question for you, if I may. Let's flash forward 20 years' time. What's the cancer environment look like then? What's the focus of research then? How do you see things changing, either more generally or more specifically in the areas that you're focused on?
Well, that's interesting, because the 20-year time period is a really interesting one, because it was only 20 years ago that we decoded the human genome. And in these 20 years, we've learned how to use a lot of that information, and we're getting much better at using that information. I don't think I have a hope in hell of predicting what we're going to be doing in 20 years' time, to tell you the truth, because things are changing so quickly. The really exciting stuff at the moment, as you probably know, is immunotherapy, and it's training the body's immune system to attack the cancer, seeing it as not self, as a foreign object, and attack it. We're doing a lot of work in trying to figure out how that works and make it happen to the tumours that aren't normally treated by immunotherapy. What we're learning now is that a tumour is a tumour because the DNA's gone wrong, or epigenetically something's gone wrong in that tumour. It's not because it's occurred in a breast or in a brain or in a colon. And so what we're starting to do now is to understand that a tumour is really the sum of its mutations, and to develop drugs specifically for the mutation. So it doesn't matter where the tumour arose, really, because it's often spread to a different part of the body anyway, right? What you're doing is you're dealing with it as a genetic malformation, aberration. And so we're getting better and better at that, learning to understand what all this genomic information tells us, what the mutations mean, how we can drug them, how we can deliver and develop other therapies that might do that job for us even more efficiently than drugs, perhaps.
Fantastic. That's probably a very positive and encouraging note to leave on. Michael, let me thank you for joining us today. We're very proud of our association with Flinders. We're delighted to hear about the good work that you and your colleagues are doing and the progress that you're making. And to the extent that we've played any role in it, we're delighted to do so. And we look forward to an ongoing involvement with you. So thank you for joining us today.
Thanks, Paul. Thanks to you and everyone at Decoded. Thank you.
Very good.